Good afternoon, everybody. You are not seeing double. I just decided that I wanted to use the same slide that I used for the introduction of the webinars as my background. And Julia was nice enough to teach me how to do it. So now you're seeing me plus the let's see, the cover of our presentation. I hope you are all doing fine, same as we are. And I want to welcome you to this third and last session for the ABCs of Plant Pathology. Again, my name is Maria de la Fuente. I'm a farm advisor here in Monterey County. And this is our third and last session where we're gonna be discussing some of the emerging diseases in ornamental nurseries and greenhouses, as well as the effect of the third factor in our triangle, which is the environment. And once we discuss those, we could see a lot easier all the proposed disease management strategies to try to avoid as much as possible the use of chemicals. And although I am not an expert on chemical usage, I'm gonna show you a couple of slides on differences in using, let's say, protectant versus eradicant fungicides. So without much ado, let's start with a survey. Let's see who's paying attention to what is being said in the previous workshops. So I want you to see if you can identify examples of diseases caused by womicides or water molds. Come on, guys, 5 more seconds. Go with your guts. Well, as you can see, most of you guess it or know it right. Remember, powdery mildews is different than downy mildews, and only downy mildews are caused by water molds. But also, I throw you kind of like a confusion thing, because crown rots and root rots are also caused by water mildew, water mold, sorry, which the ones that are absolutely not correct are the whales and blights, because a lot of fungi and bacteria can cause those two set of diseases. And you got it all right, because they don't cause galls at all. So even though they might cause crown roots, the last selection was not correct. Thank you very much. But you will have opportunities, to practice and go over again to try to make sure that we are clear with what we are trying to teach. So for this session, the specific objectives is, well, discuss the most important emerging diseases. This is kind of like weird, because some of those emerging diseases, they are already here with us. So after we discuss them slightly, we're gonna try to understand the multiple factors that affect the diseases that are caused by environmental issues, and what steps do we need to manage the diseases specifically in the greenhouse and the nursery, which are totally different approaches just because of the environmental aspects that are different. In doing that, then we will understand what are the most recommended control methods for disease management strategies. So the emerging diseases, we have the downy mildew for impatiens, or impatiens downy mildew is obviously another water mold. So we know from starting that if we manage correctly watering issues and relative humidity issues, we will have better success. But CDFA has asked us to be in the loop for these diseases that are present elsewhere in the United States, but still not in California. Another one that is not in California is the downy mildew for sunflower and rudbeckia. And then Phytophthora ramorum, we have discussed it plenty or a big deal, and it's already present in California, is the causal agent for the southern oak death and lots of other syndromes. But then we have also the Liberibacter asiaticus, which is actually a candidate for being named that name. It's a bacteria that causes the Guanglong disease or citrus greening that is also already present. Flew away all the way from Florida down into our California citrus groups. Then Ralstonia solanacearum, which is a bacterial wilt, southern bacterial wilt, is not still in California. And Phytophthora tentaculata, which is already in California, but we're gonna have a slight discussion in order to understand better this one that is particularly heavy for most of you that are in the production of natives. So here they are, how they look is they are both a common downy mildew, which you can see, especially in the lower side, the whitish mycelium that is being developed. Here with the rudbeckia, you can see very clearly the whitish mycelia or mycelium in the underside of the leaf, and later in the infection, it translates into the upper side of the leaf as a black stain or a black spot. This is how it will look also in sunflower, and the same organism attacks both sunflower and rudbeckia, and a different species attacks the impatiens. Then, oops, what happened? Then we have this one, which is very common here with all of us in California, which is Phytophthora ramorum. When you receive the taping for this presentation, you're gonna receive also a couple of papers regarding Phytophthora ramorum and the importance. So it is very critical for our natural resources because causes the southern oak death. And that's the one that if we don't take care of how we move from free areas or from diseased areas to free areas, we easily carry this pathogen in our boots or in some of the residues from the soil that get attached to our camping gears. It also attacks a lot of the nursery plants or ornamental plants. And here you can see an example in rhododendron, which also affects azalea, are very similar, camellia, and pieris. It is devastating because makes the plant also show some symptoms of wilting when it's not really a wilting, but because the affection of the spot is very close to the transportation tubes of the plant. That's why they start showing some wilting. Here is in viburnum, and it is also devastating. There is a list of that was produced by one of our farm advisors, Steve Cheswold, about the common name and the parts that are affected by this Phytophthora. And as you can see, there are a lot of them, and the list keeps on growing. So we have to be very much in the lookout for this disease and make sure that we educate people that are fans of especially hiking, because we don't want to move it to clean areas. And then this, as I told you, this is just still is a candidate. So that's why we cannot say that the bacteria is called Liberibacter asiaticus. That's the proposed name. But it's a bacteria that causes what is called as what is known as the Huanglongbing or citrus greening disease. These are the common symptoms, although you have to be careful because sometimes the symptoms resemble some deficiencies. This is the common vector, and a vector is just any organism that is capable of transmitting organism that causes diseases, mostly viruses and bacteria, but they can also vector some of the fungi, and they are usually, a lot of them, there are insects, but we have a lot of nematodes that are also vectors. We have also other fungus or fungi that are vectors of viral diseases. So a vector is just a mechanism of transmission from one disease to a healthy one. So the organism needs to acquire it and then give it back to a healthy plant. This is I love the psyllid. It's characterized by its deposits of the fecal matter resembles wax, and it creates some kind of like curling characteristics. So as I was telling you, these are all the different symptomatic things that if you take leaves separately, you can easily make sure that, you can easily confuse them with, for example, this one looks typically like either a magnesium or an iron deficiency. When the leaf looks like this and is in a newer part of the plant, let's say it's a newer leaf, and looks like this, is usually or most likely iron deficiency. When the leaves looks like this and is in the lower part of the plant, which means the older leaves, is usually magnesium deficiency. But because of this, which means there is a very generalized chlorosis and intervenal chlorosis, which means between the veins, and the veins remain green. So there are lots of things that are kind of like confusing, but the main thing is that if you see the presence of the psyllid, then for sure you are having the greening of the citrus. This is commonly happening in the fruits, and there is no confusion with any other deficiency or so. Oh my God. Okay. These two that we just discussed, they are already here. The ones that are not here, such as the Rastonia and the downy mildews, pay extra attention. It attacks mostly Solanaceae. That's why it's called Solanacearum, but can move to other plants, specifically ornamentals. And when you do a long cross cut into the stems, you can see that the bacteria occupies the xylem and phloem, and that's why it causes wilt. Here is our beautiful Phytophthora tentaculata. I have a slide that I added because when I consulted with Johanna, my colleague in Davis, the one that is in charge of the diagnostic lab, she gave me some tips. But if you're worried and you're looking, and this happens specifically or most often in the plants that are used for restoration, natives. So when you peel out in the bottom of or in the crown, you can see that the tissue remains really very remains or changes color. This is the control, which is greener. And when you peel down, you can see that it is discolored and has the necrotic tissues around it. So was detected in the US 5 years ago, and then CDFA has stated right now that it has not been recovered again in California, either in the places that we have restored or in the nurseries in the last 2 years, which is good news. It seems that it does not survive in the environment for a long period of time. She's telling me that some studies done by Norstock, they inoculated the soil with the Phytophthora tentaculata, and then after 1 month of inoculation, they were not able to retrieve it or to re-isolate it again, suggesting though that is not a persistent pathogen, as opposed to Phytophthora ramorum, which is a lot more aggressive and a lot more persistent. So in terms of regulation, Tentaculata was rated A, which is the most restrictive quarantine type of issues, just as ramorum. And now it's a B rated, so it's at the discretion of the county where you inhabit. So make sure that you contact your a commissioner in order to know what are the specific measures that you're required to take if you have a plant that might be a host for Tentaculata, which I sent you in the first, when they send you in the first video or of the first presentation, I share with you a couple of documents where they list all the different hosts that this pathogen has. So let's go with survey slide number 2. And in this specific question, I want to know if you kind of like understood what I was trying to convey about what is a vector. Not Victor, vector. And I'm trying to teach you English. Okay, let's see. Vector can be an insect carrying pollen. No. Pruning shear, not sanitized, that can, nope. Although they still can transmit diseases, but it's not a vector. So you guys, I think, got it all right. Oh my God, congratulations. So a vector is any organism, but needs to be alive, that can acquire and subsequently transmit the pathogen. And anything can be a vector. If I acquire it, I cannot transmit it because most of the diseases that I acquired or that a human being acquires cannot be transmitted to plants and vice versa. So thank you, very well. So it pays to speak slowly because everybody understands me better than when I speak in Spanish. So anyways, this is the third part of our beautiful triangle. We already discussed in depth pathogens all last webinar, and then the characteristics that the host plant needs to have, such as great susceptibility, in order for these three to coincide and have a disease. Oh, what happened? I went back. Oh, Maria. Anyway, so this environment includes multiple factors, as well as the interactions between them. But just keep in mind, for you guys, an environment happening in a nursery outdoors is totally different than a nursery or a production system in a greenhouse. So when you're in a greenhouse, you have better chances to regulate and control these factors such as temperature, humidity, light, water, soil, or potting mix or substrate, wind or ventilation, chemicals, fertilizer, and This one we tend to disregard, but is so important in plant pathology because the mere um, you know, contact of, let's say, a diseased plant with our pants when we're walking through, or our boots, may act as a carrier of dispersal spores or dispersal bacteria, and we can easily infect um, non-disease organisms or non-disease plants that are in our production crop or cropping system. So especially in the greenhouse, where we can easily control it, there is a big factor, which is the free water. We usually measure water as irrigation system, but also the water deposits on the surfaces of the leaves, and that's called free water. And it's is usually uh, provoked by all the different changes in the temperature that we have. So the relative humidity makes sure that at night, when it cools down, condensates the water and appears as dew. Or you have it outside and you have the rainwater or irrigation water, or even sprays, pesticide sprays. You use usually the water as the vehicle for distribution. So we had taught in the class before about how in 8 hours or sometimes even less, if we have this type of symptom, we can actually see the sign, because the botrytis immediately emerges from the inner tissues and produces the spores. Sometimes we can see them right away. Sometimes we don't even see them until they disperse to new plants. So we have to make sure that this doesn't happen. And this is happening, this original infection is happening because there was free water on the top of the surface. This is an diagram on how this is botrytis conidiophore, and these are the conidia. Conidias are easily released with the ventilation air or with the air outside in our nursery. And then here the conidia are deposited on the surface of the next plant, and then they penetrate. And this is what is happening. For them, usually, especially with botrytis, they do not have this particular system, which is direct penetration. This particular system emulates something like a rust. But here, if there is a lesion or if there is a stoma, so this spore germinates because there is free water. And once it germinates, remember, when it's going to penetrate for an stoma, usually produces an apresorium, and then enters, crosses the cuticle, and once inside of the cell, keeps on growing and expanding. And then when it is again relative humidity high outside, then they can come outside and produce again the spores. So if we avoid these, the free water, by increasing our ventilation, of course, or reducing our sprinkler irrigation and using more like drip irrigation or subsurface irrigation, we avoid these problems with botrytis. So here are some of the fungus that some of them require, some of them they do not require as much as water, free water, or and for many hours or minutes or seconds or whatever. So you can see that botrytis is not actually as strict as Phytophthora. With Phytophthora, you do not need any length of exposure of free water, and Phytophthora can immediately, through a wound or through an opening, natural opening, penetrate. Then we have some other rods, like in this specifically is for soybean, that it also requires very little the range of exposure. So right away you think of a mechanism of control, which is avoid the water on the leaves. And this is easily regulated not by this type of watering, but by this type of watering. And I am showing here in a greenhouse, but you can do this same thing, which is outdoors in a greenhouse, having the drip irrigation. If you want to irrigate, or you can only irrigate by sprinkler, then you have to try to do it late in the afternoon or evening, but still making sure that you ventilate, especially when you are outside, but when you are inside, making sure that you still ventilate so there is no water accumulated or attached to the surface of the plants. So air circulation and this horizontal air flow is very important in the greenhouses. This is also interesting for me because shows you all the different production tiers that you can occupy easily, but just keep in mind that whatever happens up here also might happen down here, even though they might not be very related or correlated crops. So you have to take that into consideration, make sure that when you are running the fans, all the evaporation on the surface on the surface of the plants is happening. And then that also helps keep maintaining the temperature homogeneous or uniform within the plants and in the whole greenhouse. So plant spacing is important. And when you are in a nursery producing blocks, that sometimes causes a lot of stress because you need to really pay close attention when you are doing the monitoring in order to make sure that you catch the pathogens with plenty of time. So in this slide, you can see the system where you have, usually the vents are closed in your greenhouse. So then the moist air, which is warmer at sundown, and so the relative humidity in the greenhouse starts increasing. So in order to avoid problems, just remember that if you hold the heat because you want the plants to be comfortable, you're holding the moisture too. So in order to make sure that you get it right, open vents and therefore let escape sometimes the heat. And you can usually have a system where you eat in the tables or within the tables here. So therefore, this area maintains a warmer temperature than this one, but the water that is being in the relative humidity doesn't deposit on the plants because these are hotter. So you have to open and close and repeat to avoid prolonged times of 100% leaf wetness, because if you have problems with Phytophthora cactorum or with botrytis, this only takes 6 hours with some, 2 hours with others. So be very careful. Requires a lot of planning and making sure that you program either the person that helps you with the greenhouses or your program for controlling the greenhouses. Remember, the warm and most moist air tends to go up and escape, so don't keep it closed. So let also the cool relative dry air come in and help you with the diffusion of the problem. Let's see, let's go with the survey slide number 3. And environmental factors that are managed correctly render better control of plant diseases. And these are some of them. Window and door opening, sweeping and mopping, red light and green light, water and relative humidity, and pesticide application. This is a no-brainer. So, yeah, you guys, window and door opening are not paying attention. Window and door opening will help you with controlling the relative humidity. And besides, the window and the door are not environmental factors. The environmental factors, actually, the only ones that I put there are lights, but we didn't even talk about lights, and the water and the relative humidity. Of course, if you open doors and windows, you can help control the water and relative humidity. But this is the window and door opening is not an environmental factor. The environmental factors are factors that are in the environment. So, but well done. Most of you got it correctly, got it right. So let's move a little bit into managing the diseases. So it is what we intend to do, because there is no point on knowing about plant pathology if we are not able to manage the diseases. But that takes several steps that are sometimes complicated. The first and most important is to have an accurate diagnosis. Sometimes even before that, I would say understanding what crop or what plants are you growing? Because I have people bringing sometimes samples here that they say this is a begonia, and turns out that is not a begonia. Oh, sorry, I made a mistake. So make sure that in order for you, for us and for you to have an accurate diagnosis, you also need to know the counterpart on that triangle, which one is the host. And then you can understand what are the sources for pathogens that you might have, like in all the intrinsic aspects of a pathogen's biology, such as the life cycle, the environmental requirements, and how do they spread. Sometimes by having compendium or by having some literature is a lot easier. But being that you are in an industry that sometimes you have 100 different crops in your production system, becomes kind of like cumbersome to have such a vast literature or a vast bibliothèque or library of information. But once you work with them, you start accumulating all that information in your brain. Once you understand these two counterparts together with the environment, then you can develop appropriate and effective management. Do not always try to resort to chemical management, because sometimes it's a lot more expensive and also is deleterious sometimes to your workers, or problematic to find that the label is associated with such a crop that you're having. So keep in mind also that there are a lot of abiotic causal agents, and we have another class just for that, well, specifically for the nutrients. So for us to be able to identify the symptoms that a plant might go through when it's lacking a specific nutrient. But most biotic causal agents, known as pathogens, they're small. Therefore, when you try to do the diagnosis yourself, even though with a good look, might not work. So, especially if it's the first time that you see it, you might not even know where to look at. So make sure that you take advantage of this offering that Dr. Joanna, my colleague in UC Davis, we already shared it with you, but this is the website where you go and you download this is This is her website, Dr. Joanna del Castillo. And then when you go there, you will go lower in the page and you will find the form for samples for plant pathology, where they will ask you in which county they are, et cetera, what are you growing. Sometimes if you bring it to your farm advisor, the farm advisor is the one that mails it to Joanna. And then subsequent aspects of this form tells you how to take the sample, how to pack the sample, and then how to mail it. So make sure that you use this resource, because first and foremost, it's not going to be there forever. She now has a grant that she can use to help subsidize her cost in the lab. So now we focus in strategies such as use disease-free hosts. If you are doing a lot of vegetative propagation, make sure that the mother plant is not diseased. Because if the mother plant is diseased, all your cuttings, they're gonna have developed plants with disease. So sometimes it says it's very easily said, especially when your crops are vegetables. It's a lot easier to use resistant varieties and so forth, because there's a big industry into breeding those plants. But for ornamentals, and wait a minute, even worse for what's the name? In California, we have all these natives, sorry. For natives, we don't have programs for breeding. So if they are not resistant, more likely they will never be, because we are not breeding for resistance. And besides, that defeats the purpose of being a native. But you can always make sure that then you you manipulate the rest of the environment in the perfect way. So even though they are susceptible, you control the pathogen before it even gets exposed close to the plant. Then the cultural practices, make sure that they are favorable to your plant, to your crop, not to the pathogen. Like this one, it's a big no-no, because it's a lot easier for us to irrigate like this. It is also a lot easier to the pathogens to spread and enter the host with this. So try to use sometimes a lot more expensive means of watering, such as sub-irrigation or drip irrigation, but a lot more secure and safe for your crop. Vector control, that goes without saying, but sometimes I have to insist. For example, if you control the weeds around or not control the weeds around your greenhouse, these are harboring insects, as well as sometimes the same pathogen. The pathogen can enter through any of your ventilation systems, or when you open the door to come in, it follows you with the wind. So try to make sure that you think of all these other issues that are probably outside of your production area, but as important as your production area. People move from one place to another without assuming any sanitation treatments. And now I love the way that we are living right now, because sanitation is so important for our human transmission of diseases. Well, think in the same terms for plants. So let's see, for example, how difficult it is to have a pad that where you can go through and always sanitize your shoes. I know people that have series of greenhouses one after the other, yet they don't have paths. Leaving one greenhouse and coming into a different greenhouse needs to be protected. Always walk against the titration, let's put it that way, or the amount of pathogen possibly present gradient. So if you're gonna work in your greenhouse, always move from the youngest plants towards the oldest plants. Because if you do it in reverse way, the oldest plants, most likely, they already have a hidden or not so hidden pathogen that you are gonna be spreading to the rest of your houses. Then the tools. I mean, how difficult it is to buy Clorox and then just disinfect your tools with the Clorox that you buy in Costco and whatever, and then make sure that you submerge them for at least 10 minutes before you use them for pruning or moving plants, etc., etc. I love this picture because people, they are taking care of accumulating all the debris that they produce in the greenhouse, sometimes just outside of the greenhouse, because they want to compost it and use it. Very interesting and very nice to reuse it in their potting mixes, but creates a source for inoculation, for infection. And also, if any of your living plants that you have there, they are also hosts for the diseases, the risk of having them is imminent to having them inside the greenhouse. As I was telling you, I mean, this is a seedling tray, and you usually go and pay attention, but do not really pay attention, because in this circle, although I should have put the circle a little bit, I mean, I can see this area where, I mean, I cannot see it, obviously, and without glasses, but look, this plant is already dying. And if you irrigate with sprinkler irrigation, there's the water that is splashing from this cell goes to the others and goes to the others. So you need to be really observant. And then when you do a sanitary cleaning, so you remove this one, but also you remove the chunk of all the neighbors that are surrounding, because for sure the pathogen already jumped. Another easy way to do sanitation is with the heating with vapor of your soil. Although nowadays I have not seen very many greenhouses where they still plant in the soil. They usually have substrates, or they are outdoors. When you are outdoors, it's a lot more difficult to do sanitation of the soil via vapor. And sometimes this is interesting when you produce your own substrate, because you need to make sure that you sanitize it to the level of need that you want it. It's very easy. You only need 120 Fahrenheit to get rid of the water moles and nematodes. Some fungi also die. But most plant pathogenic bacteria, as well as all the slugs that are a terrible pest, all the worms, and most fungi, they die between 140 and 160 Fahrenheit temperatures for 2 hours. You have to kill viruses, for example, and some weed seeds with temperatures above 200 Fahrenheit. It is sometimes very difficult to achieve that. You need to have a specific autoclave or a long autoclave, which I have seen in some of the nurseries that I've worked with. But Regulating the temperature that you administer to your mix before planting is important in order for you to make sure that you don't have pathogens. And then a little bit touch on the fungicides. So because most of the pathogens or fungi, they are using this free water that forms on the surfaces of the plants. So making sure that exactly in that area is where we catch them and break the cycle. It is how it works for a lot of protectant fungicides. So if we go and spray this beautiful pink protectant fungicide, which is gonna happen is, okay, there is still liquid, so the spore is gonna engorge and germinate. But once it germinates, the fungicide is gonna kill it. Therefore, you don't have penetration and you don't have internal infection. There are some fungicides specifically with powdery mildews that can work as eradicants. That means we already have the infection and then we are seeing some of the patches of spores emerging from the surface. It could be the upper or lower surface, especially with powdery mildews. So then I apply, in this case is piperylene, which is one of the eradicant fungicides. And this is what happened in 2 days. So I apply here, and then in 2 days, I can see the spores are dying. And in 5 days, all the mycelia that is on the surface of the plant is also dying. So I stop the spreading of the disease. I mean, if I would have taken a picture of, or 10 days later, you would still see a spot in your plant. You know, you still can see that this is necrotic tissue because those cells already died. But at least you stop the dissemination or the spreading of the disease. When you work in ornamental, sometimes the customer has very strict quality desires and might not tolerate spots on your plant. In this case, it's a rose. So if this is a leaf that is going to be removed because it's a cut flower, then no problem, no issue. That's why it's used. Okay, so let's just go quickly a little into a disease summary. Disease strategies for control. My recommendation is always use integrated pest management. Do not focus on a single method. And the good detection, early detection, and if you can already identify it, ID is the best way. Use as much as you can the resistant varieties, and then all the sanitation techniques or methods possible. Once in a while, at least have a year-end routine cleanup. But when you remove all the debris that is fallen and all that, bury it far away from your greenhouse, far away from your nursery. Do not leave it piling over there because it's a source, a harboring space for all the pathogens. Well, the removing of all the disease and dead plants goes without saying. Clean tools, clean your hands, clean pots, flats, benches, everything, superficial disinfestation at all times. And if you want to sterilize soil or potting mix, you can use heat, either dry or with steam, or use some mechanism called solarization, where you just expose it to the sun. And with a thermometer, you measure what type of temperature. You put it under plastic bag, you create a greenhouse effect type, and the vapor that generates kills the pathogens if you reach the temperature that you are aiming to. Improve always the greenhouse environment, reduce relative humidity as much as you can just with the horizontal ventilation. Do not overcrowd your plants, although, you know, with cells that are 1 centimeter, square centimeter, they're crowded. The beginning of your plantings. Alter different cultural practices. So if you irrigate with sprinkler irrigation, next season, irrigate by soft irrigation. Of course, control the weeds and the insects inside and outside, and use, if you want to use chemical control, biopesticides is the best option, and then regular chemicals, which I do not know much about them, is your last resort. There is a beautiful site in the university, which is, this is the web, http://ipmucdavis.edu, where you can Google, search by crop or search by pathogen or insect inclusive. And I invite you to go and try to look. I mean, I am sure that there is very little information, not much information about native nurseries, but relatives of native plants might be listed there, and that gives you an idea. The university also has many references that might be of help for you. This is the other website where they have the catalog. They have visited because they have a lot of free publications, although most of the books, of course, are for sale. This is an interesting, very beautiful book that speaks about floriculture and nurseries. And I use it as a resource in the class for ABCs of pests. So you guys make sure that if you're interested, just glance through it. This is when you want to make sure that you have a business management manual as well as a production manual, specifically for container nurseries in the outside, although you can, it also touches a little bit on container production inside. And this is a brand new publication from one of my colleagues that I work a lot with nutrients and fertilizers, and it's called Water, Root Media, and Nutrient Management for Greenhouse Production. But also, American Phytopathological Society, and being a plant pathologist, has several compendiums, which I think the plural should be compendia, of diseases for ornamentals. And here I included the citrus because some of the citrus are also used as ornamentals. Go to their website, just American Phytopathological Society, and look for some of those compendia that might be of use for you. And did we discuss the most important emerging diseases? Some of them are not emerging, they're already here. And did we understand the multiple factors of the environment that affect diseases? One of the most important, water and relative humidity, as well as surface water. And understand the steps that you need in managing the diseases for the greenhouse and or nursery, and some of the management strategies and the most common control methods. I hope we did it. So, thank you very much, and I'm open for questions, and I know that Julia is there, so if she can help me out, I would appreciate it. Thank you.
Yeah, absolutely. Thank you, Maria. First off, we had someone is asking if you can show the slide again about how to submit samples for pathology testing.
I mean, I actually didn't show the slide how to submit. It's just the slide that once you go to this website, to this website. This is the cover that you're gonna see, and down you will see a form that you download and print out, so you submit the sample. But I didn't print the whole form. The whole form is like three times longer than this one, where they tell you how to sample. So where they're gonna tell you how to sample is in here, in this website. How to sample, how to preserve the sample, how to mail the sample. And this is my colleague, Joanna del Castillo. And as I told you, she is right now working with a grant that provides the service for growers for free. So take advantage of this opportunity. We used to have here in Monterey County a plant pathologist that also receives samples. I don't because now we don't have resources in the lab, but and he retired from the university and became a private consultant or works for a private, but there are many labs. By the way, I think this is something that I should send you, Julia, so you can post it for the many labs that you can send your specimens, your samples for diagnosis or for other purposes, let's say soil samples or for analysis, and that they are kind of like recommended by the university. We used to have a lot more support for providing those services to the growers, but the constraints with budgets in the state and the constraints of budgets in the counties and all that has prevented us from being at that level of service that we used to have in the past. But you talk to your representatives or your senators and yada yada, and just tell them how important it is for cooperative extension to have total support so we can support you. That will be very good.
That'd be great. Yeah, absolutely. All right. So yeah, Maria, if you want to get me that website, I can certainly include that in the follow-up email to folks.
Thank you, Joanna.
Sure. Our next question, if nematodes are killed, no problem. If nematodes are killed at 120 degrees, do you recommend solarization of outdoor planting areas? They're particularly concerned with root knot nematodes.
Oh, root knot nematodes, yeah, for sure. The only one that you need to be a little bit careful is the cyst nematode, which is also one where the female, when it's gravid, is rounded, because they have a specific mechanism of encapsulation and they resist higher temperatures. The female acts like a cocoon where all the eggs, so the female dies, but all the eggs within survive. So, but I haven't seen the cyst nematode here in California yet, but the meloidogene, yes, and it will die, female eggs and babies at 120, make sure 130 Fahrenheit.
Okay, great. Question, is there a resource for effective bioherbicides specifically?
Man, I can get you that stuff, biopesticide, the biopesticide list. I do not know it by heart because I consume them actually very little. But of course, there is a lot of resources for biopesticides. I know a little bit more about biopesticides for controlling insects. There are a lot more popular and more common, and I am assuming that there might be kind of like the same distributors and the same type of companies, but I'll make sure that I post that and it's sent to you when we send you the information about the closing of this webinar.
Great. Perfect. Do you have any resources for labs that do pathology testing on irrigation water?
Yes, I will send you also all that in the resources. So it's gonna be posted.
Perfect. We had a few questions that went into the chat box. So let me switch gears and pull those up. You did not talk about any latent infections, diseases that initiate in the greenhouse. They don't show symptoms, but once the plants are placed in gardens or fields, then the disease is expressed with symptoms. Are there any treatments for possible cryptic diseases or latent infections?
Yeah, those are mostly, or the way that I know them is mostly through viral infections. I honestly have never seen, heard, or view, but I would be very interested if you guys communicate with me, and I'm gonna go back to my last slide so you guys can send me emails, because that problem is very common with viral diseases, but not so much with pathogens such as bacteria, nematodes, or fungi. So make sure that if you have that situation, email me and I can get you some information specifically to that. And that is important because, especially when you are in the business of producing seedlings, logs, or starters, or however you want to see it, you have to make sure that you provide kind of like a certificate of insurance to your customers. And sometimes it's very difficult, especially as I said with viruses, because in the viruses, you don't have the prevailing environmental qualities that are needed for the expression of the virus in the plants while you hold them in the greenhouse. So if you are having such a problem with other pathogens, please let me know. I would be very, very interested in hearing what is the case and what is the issue. What is a lot more common is when you, as a greenhouse grower, buy seed that is not certified. And usually the seed is contaminated and carries one or two or many pathogens. And therefore, that's a lot easier to argue because, it's impacting your whole business operation.
Yeah, absolutely. We had another question in the chat box. Does steam instantaneously sanitize surfaces between crops?
Does steam instantaneously what?
Sanitize surfaces between crops.
Oh, no. And then so remember, the steam is just water heated at a very high temperature. Mm-hmm. But in order for that to work, is like your microwave, very similar to your microwave. When you put something to be heated in the microwave, needs to have some water content, otherwise won't heat it up as required. So surfaces with sanitized with vapor, first and foremost, is extremely expensive. And then if your surface is, let's say, wood, the wood also harbors a lot of a lot of microorganisms that are not necessarily pathogens for others or for the wood, that are not killed. So let's say in trying to disinfect wood, you need to use other type of substances that also permeate in the inner part of the wood as opposed to the outside of the wood. Same thing like with the Clorox. Clorox sanitizes only superficially. That's why when you have a mold in your bathroom, if you spray the Clorox, you will kill it superficially right away. But that doesn't mean that in 2 more weeks or 3 more weeks, it won't come back again, because you cannot kill it in the depth of the wood or of the surface, which is usually wood in your house. So think about all those issues. Of course, if you have metallic surfaces, yes, easily and readily sanitized by vapor because it's only in the surface. They cannot go in, they cannot penetrate, neither the vapor nor the pathogens.
Okay, great. Another question.
Sorry, I just wanted to add something else. For
Oh,
example,
yeah.
that's the same situation when you sterilize tools. If the tools, they're dirty, they still have some soil, it won't sterilize them completely because the Clorox is just a surface disinfectant. So the soil particles or the substrate particles can still harbor and hold pathogens. You need to make sure that they are clean before being disinfected.
Okay, and kind of along those same lines, we have a question about if you're wanting to reuse pots and seed trays, how long does the bleach need to stay on the surface?
Well, it's not matter of the bleach staying in the surface, it just matter that there is no organic matter in that surface. So if you want to reuse your pots, make sure that they are perfectly and absolutely clean. Now, sometimes it's a lot more expensive to clean them because the water is a lot more expensive sometimes than the disinfestation. But if you submerge them and leave them for a while, you can assure, I mean, I never done the experiment, but I am assuming that with a couple of minutes submerged in the Clorox would be enough as long as they are clean.
Gotcha. All right. That's all the questions we have right now. If anyone else has more, feel free to add them. And you guys, just to let everybody know, we'll be getting an email within a week or so with follow-up information to this webinar. So with an evaluation form to fill out and with some additional resources and all of the recordings from these. So be on the lookout for that. And it looks like we have a few more questions that have come in. Do you use Clorox straight or diluted?
Oh, it's diluted, always diluted. Usually the proportion is 1 to 10 with a 1 to 5 or 1 to 10, sorry, I forgot exactly. If you buy the Clorox, the one that you get in Costco, I think you dilute it 1 to 10. But if you buy easier hypochlorites or industrial hypochlorites, the dilution is slightly different or is different. So
is that something you can find on the label? Yes, I would assume, yeah.
And I'll send it to you, all that information. I'm making notes here.
Okay.
All the information that they are requesting.
Perfect. One other question that came in, I want to know what the high temperatures that nursery trees, nut crop, or stone fruit can be tolerant of.
Oh, you mean you want to treat the root stock? You want to treat the tree? I mean, I do not understand very well the question. Or is he asking, or he or she asking about environmental temperatures for production? Can you re-specify your questions, please?
Yeah, maybe they'll type their question a little bit better for us. All right.
And also I wanted to take this opportunity to remind you that next Friday, we will start this same training, but all in Spanish. So if you have foremen or employees that you want to promote into their professional development, feel free to email us so we can send you the information how to register them.
Absolutely. Good reminder. All right, we can give it a few more minutes and see if that person puts in more. Oh, here's one other question. Is it okay to reuse a pot without cleaning it if the plant that was in there did not show any signs of disease?
Um, my best advice is no, because as you said, you don't even know how long it took for that pathogen is might be still growing into the remaining of the of the media that is still present. Is not recommended, and you will avoid a lot of issues by making sure that you use clean pots or clean trays.
Okay. And let's see, it looks like we got some clarification. I think if high temperature can limit the growth of latent infection pathogens, that the host can tolerate it too. That's their concern.
I will do some research about it because no idea. I never worked with fruit trees, but I know that they start like babies, like in any nursery. So I will do some research and post it in the material that Julia is gonna be sending you sometime next week.
Okay.
I apologize.
That's okay. You can't know everything.
Yes. That's what I tell my daughters all the time. Excuse me.
Do you know of any places or centers that recycle plant pots?
Oh, if you want to recycle plant pots, contact the master gardeners in your local county, and they will actually go and grab them for you. Because master gardeners, they do a lot of practicing of their skills with donated plant pots. And if not, here in Monterey County, we take them. We are located in 1432 Abbott Street. If you live close, ship it our way or bring it our way.
Absolutely. Great. All right, there's nothing else open at the moment.
Well, I want to thank you for bearing with me. Sometimes I know it's kind of like difficult to, to be with somebody that doesn't have the same accent that you do, but I make sure that at least it's written in correct English and that you can follow us. And I am so thankful to have had the opportunity to be acquainted with you guys, although you saw my face and I never saw your faces. I hope you learn a lot, and my email is there. If you didn't, do not hesitate at any time to email me. And I want also to thank Julia for all her support and all her, she's a millennial, obviously, so she knows a lot about these issues that I don't. But she was just a pleasure to work with and to have here supporting our educational efforts. So thank you again for attending this series of webinars, and hope to see you or hear from you soon. Thank you.